screen. All right. So, hi everybody. My name is Karis. I am a graduate student here at Northwestern. I'm in our Driscoll graduate program, so that's our biomedical life sciences program. I'm actually on the downtown Chicago campus because of that, so I'm right by the hospital. I am in the lab of Navdeep Chandel. So what my lab studies is broadly, we study metabolism and the mitochondria, which you may or may not be familiar with as like the powerhouse of the cell. That's what its claim to fame is. But I specifically study mitochondrial dysfunction and metabolism in Parkinson's disease. So I wanted to just talk to everybody today about how do we study and how do we treat Parkinson's disease. I think a lot of people, Parkinson's disease is well known enough that people know kind of generally what it is, but I think a lot of people don't really know how exactly we study it in a lab, and then also what treatments are available and how those treatments work. So that's kind of what we're going to go over today. Feel free to ask questions throughout. You can ask them in the chat. You can just unmute and say it out loud. Feel free to interrupt me. This is kind of meant to be more of a conversation just so that you all can learn more about the brain and how it works and what's going on when it's not doing exactly what it should be doing. So with that, I will get out a pointer and I'll get started. So just a little bit of background on me, just so you can know who I am, what my educational background is like, and also see me without a mask for a second. So my name is Karis D'Alessandro. As I said, I grew up in Tulsa, Oklahoma. So that's me. And then I moved to Baltimore for college. I went to Johns Hopkins. I got my bachelor's degree in neuroscience there. After that, I moved to Nashville for a year. I worked at Vanderbilt in the lab of David Calkins. I actually worked in glaucoma research there. So if anyone has any unrelated questions about that, I can do my best to answer. It's been a couple years since I've been there though, so I might not have every answer. And then after working there, I moved here to Chicago to go to Northwestern. So now I'm a second year PhD student, like I said, in the Driscoll graduate program here at Northwestern. So just a general overview of what we're going to talk about. First, I'm going to talk a little bit about the basic fundamental neuroscience background you need to kind of understand what's going on in Parkinson's disease. Then I'm going to talk about a couple different research methods that we can use in the lab to study Parkinson's disease. And then I'm going to talk about the treatments we have. Unfortunately, we don't have a lot of treatments. There's two main treatments I'm going to talk about, which is L-DOPA, which is a pharmaceutical treatment for Parkinson's disease. And then I'm going to talk about deep brain stimulation, which I personally think is like the coolest thing ever and hopefully can convince you of that too. So kind of just starting generally, one of the things that is important to understand when we're talking about Parkinson's disease is what do the cells in our brain look like? Because we have a very unique type of cell in our brain that we don't have anywhere else outside of our nervous system. So the cells in our brain are called neurons. So here's a nice little picture of a neuron. One of the important things to note in this slide is just that we see that our brain communicates using impulses, electric impulses. And what this is showing us is that the impulse goes in this direction. So that's what this arrow is showing us down here. So that's really kind of the main thing that I wanted you to see on this slide is just the general shape of a neuron. It's like kind of a linear looking cell. And then our impulse goes from one side to the other. And it goes to the end here, which is called axon terminals. There's some other things going on in this slide. If anyone has any questions, I can talk about it. But basically, this is just a perfectly designed cell that is meant to efficiently move an electric impulse from one end to the other. So that's a neuron. So then another thing that's important to understand when we're talking about Parkinson's disease is how do neurons communicate with each other? So this kind of, you'll recognize this neuron from the last slide. So remember how I said the impulse goes from this part of the cell, this big, like it's called the soma, the cell body, to the axon terminal. So the reason why it does that is because then that axon terminal connects to the next neuron. So that allows for one neuron to communicate with another. So there are kind of two ways that cells communicate with each other, but they work together. So one of those ways is using neurotransmitters. So that's what these blue dots here are supposed to symbolize. So in the case of Parkinson's disease, we are talking about dopaminergic neurons. So that just means that these are neurons that use dopamine to communicate. What neurotransmitters do is they basically tell the next cell, okay, you need to initiate what's called an action potential. So you can see that up here in this corner. And what that is that's just the electrical impulse that I was talking about. So that just is a general idea of how neurons work, how neurons communicate with each other. Obviously, it's important that neurons communicate with each other in the correct way in order for our brain to function correctly. And that's going to be emphasized here on this next slide. So kind of what I'm showing you here is the substantia nigra. So this is a human brain from a couple different sections. So just so that you can kind of get your bearings here, this first slide would be, or sorry, the first brain on your left would be if you cut it, like if you cut my head this way, it would give you this perspective that you're getting there. The second one would be if you cut my head like this, like up and down, you would get that perspective. And then this last one is if you cut basically like from the forehead to the back, like just like this, you would get that perspective. So in Parkinson's disease, we specifically see problems with the neurons in the substantia nigra. So this red is just kind of showing you this area is the substantia nigra. So I'm just kind of trying to orient you a little bit so you can see what part of the brain is involved in Parkinson's disease. The substantia nigra also, so obviously one of the main symptoms of Parkinson's disease is movement problems. So the substantia nigra is part of a big network that's on this slide that is involved in movement. So this is kind of just showing you, like I said, it's really important that neurons are able to communicate with each other the way that they're supposed to in order for our brains to function the way that they're supposed to and for our bodies to move in the way that we want them to. So this shows us all these connections. So down here is the substantia nigra that I was talking about. You can kind of recognize this shape from, from here. We've got the same shape of the substantia nigra right here. So that just kind of orients you a little bit, but we see that it's connected to all these other things. All these other things aren't necessarily important for this talk. The only thing that's important that will come up later when we talk about deep brain stimulation is this, the STN. That's the subthalamic nucleus. You just need to know it as the STN, but that's actually where during deep brain stimulation, that's where they will implant electrodes to try to alleviate symptoms of Parkinson's disease. But basically what I'm showing here is just that there's a really big circuit. All these regions play a role in initiating movement, which is, I think to me, I find that to be part of the beauty of the brain, that it's kind of super integrated across many different parts of your brain, and they all kind of have a place and a role, and it's important that they're all functioning properly. And when one of them doesn't, like for example, in the substantia nigra, it can lead to something like Parkinson's disease. But what's actually important to mention here, and I think speaks to how awesome our brain is and how plastic it is that it actually requires the loss of about 90% of neurons in the substantia nigra before anyone would start to notice symptoms of Parkinson's disease. So our brain has a really amazing capability to compensate for loss of cells. And it's just a super robust organ. And I think it's really cool because I study it. So now on to a little bit more specifically about Parkinson's disease. So I've kind of covered most of this already. So in a healthy neuron in the substantia nigra, which is what we're seeing here, dopamine would be used as the neurotransmitter, and there's plenty of it, and it's able to relay any message it needs to using dopamine because it's got more than enough. The cell that wants to send the signal and the cell that's going to receive the signal are both alive, so it's no problem. The reason you can see this coloration right here in the substantia nigra is actually something really cool about our bodies, I think. So in the process of dopamine being made, one of the byproducts is melanin. which is what causes, color in your skin, color in your hair, that's like melanin. So it's really cool because if dopamine is being produced in the right quantities, you will start to see melanin in this part of the substantia nigra. So in someone who has Parkinson's, actually, in their brain, they don't have as much melanin in the substantia nigra. So it's actually a lot lighter. It should be really dark. It's basically black in people that have like a healthy functioning substantia nigra. But then in others with Parkinson's disease, you start to see a loss. And that's due to a decrease in dopamine signaling. You can basically use the color and the presence of melanin as like a proxy for whether or not dopamine signaling is happening. And this down here is just kind of emphasizing the fact that, so these are all pathways that use dopamine. I'm sure you're familiar with the fact that dopamine is associated with like motivation and emotion, but it also does play a very big role in movement, as we can see in Parkinson's disease. So now I'll talk a little bit about the research approaches to Parkinson's disease and how we study Parkinson's disease in the lab. So we've got all sorts of options for studying Parkinson's disease. So we can use what's called in vivo models. That just means in a living organism. And believe it or not, we can study Parkinson's disease in organisms as small as flies. So this is called a Drosophila, is the Latin name for it. And you can do things like knock out a gene in the Drosophila and then see if they have climbing deficits. If they can't climb very well, that would be like considered symptomatic Parkinson's disease for a fly. Then we can also use mice. This is what I use. I use mice. Rats are also something that are used relatively frequently for Parkinson's disease research. Monkeys, and we can also use people. There are a couple different ways that you can use these animals, particularly as models for Parkinson's disease. So you can do what I kind of just described. You could do a genetic manipulation. So there are some genes that have been identified as being related to Parkinson's disease. However, the vast majority of Parkinson's cases are what we call sporadic, which means that there is not a genetic mutation associated with it, and it just occurs sporadically, as the name would suggest. And so to mimic that, another option that a lot of researchers will take in modeling Parkinson's disease is they will use a neurotoxin, like rotenone or MPTP or paraquat. Paraquat and rotenone, I believe, are both used in pesticides. So there are actually human cases of Parkinson's disease that have been tied to previous exposure to those things. So those are all the different models that not all of them, but those are some of the animal models that we can use to study Parkinson's disease. I think a lot of people don't realize that we can use all these animal models and things like that. So I just kind of wanted to show you all the breadth we have in terms of how we can study Parkinson's. Aside from just those animal models, we also have, cells. So actually, this is perfect. So we have in vitro models that we can use. So in vivo means in a living organism. In vitro just means, I believe it means like in glass. And so it just means on a Petri dish, basically. So we can actually use cells. So this is the SHSY5Y cell line. So this is a cell line that's used pretty frequently to study Parkinson's disease because these can turn into neurons that express dopamine receptors. So obviously that'd be important for studying Parkinson's. I'm seeing something in the chat now and I'm looking at this. Okay, interesting. So Carla asked, my sister who's 65 has both MS and now Parkinson's. Do you have any idea of the % of people who have both diseases? And then also, is it possible to get a recording of your talk? So we are recording this talk. It's going to be posted on our YouTube channel. I'm not completely sure what the link for that is, but maybe if Suyag, if you could just like throw the link into the chat so that people can have it. Obviously, it won't be posted right away, but then as soon as it's posted, you should be able to have access to it. So talking about MS and Parkinson's, I don't know how many people have both, but I can go back and show you essentially what's happening in MS is, so this is the neuron. What's happening in Parkinson's is we see cell death and we don't know why. And then we also, well, I would like to know why, because that's what I'm studying. And then we also see a loss of dopamine receptors on dopaminergic neurons. What's happening in MS is this is what's called a myelin sheath. Basically what it does, if you think about this like a wire, all your wires have, here's a wire, right? Like there's something on the outside that protects, that does a couple things. First off, it protects the signal, but then also it helps make sure the signal's really fast, right? Because it's making sure that none of the electrical current is getting lost and that it's all able to go where it's supposed to go as quickly and efficiently and with as much of the signal left as possible. So that's what the myelin does. Basically what's happening in MS is the myelin is breaking down due to like an immune response to the myelin that's causing it to break down in the peripheral nervous system. So I actually don't know the percentage of people who have both MS and Parkinson's disease, but that's just kind of an explanation of what's going on there. Those are different functions of the neuron, but they're both neurodegenerative diseases for sure. All right, so that was a great question. So now going back to the cell line. So yeah, this is just kind of what cells look like on a dish. You can kind of start to see, you know, they kind of look like neurons. They've got that same long shape that I was talking about. All right, so now I'm just going to talk briefly about the treatments for Parkinson's disease. So unfortunately, we don't actually have a ton of treatments for Parkinson's disease. We had for a while just L-DOPA, which I'm about to talk about, which is short for levodopa. I think it was discovered in like the 50s or the 60s. It's been around for quite a while, and until very recently, until DBS came out, it was really the only treatment that people had for Parkinson's disease. So essentially what L-DOPA is, it's a pharmaceutical intervention for Parkinson's disease. And what it does is basically just produces more dopamine. So L-DOPA is actually a precursor to dopamine. So you can kind of see this here. So someone takes L-DOPA, they have L-DOPA in their bloodstream. It's able to cross something called the blood-brain barrier, which is actually really cool. There's a lot of drugs that can't cross the blood-brain barrier. The blood-brain barrier, which I'm just going to call the BBB now, because if I say that too many times, I'm going to say it wrong because that's a really hard phrase to get so many B's in a row. But the BBB basically is an immune protection for your brain. So it protects, you know, infections that you get in the rest of your body from going to your brain and that sort of thing. But also, unfortunately, when we're trying to develop drugs, that also means a lot of drugs aren't able to cross that and get into the brain. But L-DOPA is, which is good because it means that then we can use it to treat Parkinson's and things that are going on in the brain. So this schematic is kind of just showing you L-DOPA comes in the bloodstream, crosses into the BBB, which is then means it's in the brain. So now we're in the striatum, which is a part of the brain that's associated with movement. That's kind of where like all these things I showed you in that circuit diagram are. So very important for Parkinson's disease. And then neurons are able to convert it into dopamine. So they're able to restore that dopamine signaling that I said in previous slides is kind of what's lost in Parkinson's disease. So that's just a little bit on how L-DOPA works. But the thing is, L-DOPA doesn't necessarily work forever. A lot of patients will start to show some resistance to the effects of L-DOPA. It only works for a certain amount of time. And so that's why DBS kind of was pioneered as a possible treatment for Parkinson's disease. So I'll talk about that next. So over here on the right, we've just kind of got a schematic for what DBS looks like. So what happens during a DBS implantation, DBS stands for Deep Brain Stimulation, is electrodes are placed into the subthalamic nucleus, which is the STN that I mentioned before. So it's part of that circuit, that's part of the substantia nigra. There's a battery and a controller that's placed into the chest area. And then there's wires that connect up to the electrodes. When I was in undergrad, I was considering being a doctor instead of a scientist, and I shadowed a neurosurgeon actually, and I got to see a couple procedures during which they implanted these electrodes. And I remember one of the surgical nurses telling me there are, DBS implantation is the third most beautiful thing that you can see happen in a hospital. You've got births, you've got organ transplants, and then deep brain stimulation is definitely rounding out the top three. So it is super, super cool to see. I'll play a video for you guys over here on the left. So on the left side, we've got this gentleman with Parkinson's disease. You can see what he looks like before he turns on the deep brain stimulation, because like I said, this like battery pack and controller is implanted in the chest, so you can actually turn it on and off. So you'll see it, he's got it without, and then after we turn it on, we'll be on the right. So I'll go ahead and play it. I'll probably play it a couple times because it's kind of quick, but it's really cool. So on the left, we can see, the Parkinsonian, the tremor in his hand, and we can see, and then on the right, look at that. He's doing the movement, not totally perfectly. He's off a couple times, but I mean, significant improvement. So I'll play that again so you can see it. So again, on the left, that's when he doesn't have his deep brain stimulation. And then on the right, he's got his deep brain stimulation going. You can find videos online of people literally turning it on for you as they're sitting there, and you could just watch. I mean, within seconds of turning it on, it starts to work. As cool as deep brain stimulation is, we don't actually fully know why it works. There's a couple theories as to why it works. Obviously, what's going on is it's disrupting the electric current signals that our brain is sending. One of the theories is that, so in that circuit diagram that I showed you, there's obviously a lot of different parts of the brain that are associated with movement and associated with Parkinson's disease. So in Parkinson's disease, what we see is that all these parts of the brain, all their electric signals actually sync up, which is not normal. So normally in a healthy motor circuit, like I showed, there's actually a lot of very asynchronous activity. It kind of just looks random and sporadic. Whereas in patients with Parkinson's, it starts to look very synchronous. So potentially the way that deep brain stimulation is working is that it's actually causing that sporadic activity that we see in a healthy motor circuit as compared to that like synchronous activity that we'll see in somebody with Parkinson's. So that kind of concludes everything that I had planned on going over with everybody. We can watch it one more time just so that you guys can see how cool it is. And then feel free to ask me any questions that you have. You can put them in the chat, or you can unmute yourself and ask. I love talking about the brain. I love talking about all of this stuff. So feel free to ask me anything. All right. Okay, I've got a question in the chat. How does dancing help Parkinson's patients move? That's a great question. I don't know the answer to that. I mean, my thinking would just be that practicing movements and practicing coordinated movements is definitely helpful in Parkinson's patients, but I can't give you a super in-depth answer to that. I hope a doctor should be able to, and also, I mean, if it's working, it's working.
Great, thanks for the really great talk, Karis. Such an important topic and really cool stuff to be doing research on. I had a question about kind of like the long-term effects of deep brain stimulation. So, you mentioned that it's like an implanted surgical device that then presumably they live with, right, for a long time. So and it seems like, when it's off, the symptoms still persist. So kind of like, what are the long-term effects on this? Is there like a tolerance that builds up, or is it just like it always works no matter what?
Yeah, so that's a really good question. So an interesting thing about the deep brain stimulation is that it actually, so with L-DOPA, that drug that I talked about, so first off, to be eligible to receive DBS electrodes, you have to actually L-DOPA, you have to be responsive to L-DOPA. Some people with Parkinson's are not. So L-DOPA doesn't work for everybody. And like I said, it stops working over time. So you have a specific window of time during which you can do DBS if that's something you're interested in. It has to be when L-DOPA is still working. The phrase that they use is on time versus off time. So L-DOPA has on time, that's when it's working, and off time is when it's not. So in a patient, they will have a certain number of on-time hours or off-time hours every day, basically just like when the drug is working and when it's starting to wear off. So in order for DBS to work, you still have to have on time and still have to have a certain amount of on time. So with something like DBS, you would still be taking L-DOPA. You would still be doing all those sorts of things. So once those other effects wear off, DBS might not be as helpful. But I mean, that being said, I would also say that DBS is relatively new. And so because of that, we don't fully know long-term what all the effects look like. So let's see. Okay, so I've got a lot of questions in the chat about DBS. Okay, what percentage of DBS patients have unintended side effects such as speech issues? I am not completely sure to the answer of that question. I am not a neurosurgeon, so I don't do actual DBS implantations or anything like that, but I can tell you the implantation is actually done while the patient is awake. Specifically to try to avoid any of those types of side effects. So they will, as they insert the electrode, like have the patient speak and have the patient do all these things and be like, can you move your hands for me? Can you move your feet, to make sure that they're going to the right location and not interfering with, a speech area or anything like that. How long do patients with Parkinson's disease usually live? I'm not sure the exact number, but I can tell you that it has increased over the years as we've come up with better interventions. We also just have a lot better treatment for people with Parkinson's disease now. And then also, do electrodes stay implanted inside? So yes, they do. So the electrodes would stay implanted, and then also the battery pack in the chest that I talked about, those would stay implanted. The battery pack will have to get replaced every few years. But other than that, yep, the electrodes just stay hanging out. What research is happening for more of the mental symptoms? Okay, that's actually a really interesting question. So that's something that I think recently researchers and scientists have started to realize that Parkinson's disease is not just motor related symptoms, that there's also a lot of cognitive and mental symptoms associated with Parkinson's disease. So there's a lot of research recently, I think, that's going into that. Unfortunately, science moves kind of slow. And so once something starts to become a topic of interest, it takes a while until that becomes something that would be, really well established in the field. But I think that there's a lot of research that's going into the mental and the cognitive effects of Parkinson's disease. For example, in my project, we actually study we actually assay for depression and anxiety and those sorts of things when we look at Parkinson's disease. And then there are also studies that I think focus specifically on the mental and the cognitive side effects or symptoms of Parkinson's disease. Some insurance plans cover DBS. Any idea of the cost? I have no idea of the cost, and I'm not sure if all insurance plans would cover it. I would assume most probably would cover at least part of it, but again, I am not sure at all. You would have to verify with your specific insurance to make sure that it does.
Great. Thanks so much for answering all those questions. Yeah, so if there's no other questions, then we'll go ahead and wrap this up. So thank you all for coming. This talk will be posted online. It'll take a couple of days for that to be set up, but then we'll send it to Tim and he will pass that on to you. So thanks again, everyone, for coming, and enjoy the rest of your day. Thank you, Karis and Katie and Nicole.
Thank you. Great presentation.